This video is about CRISPR, the gene editing tool. So what does CRISPR actually do? Well, it's enabled scientists to essentially cut and paste genes quickly and effectively. CRISPR itself is an acronym which stands for Clustered Regularly Interspaced Short Palindromic Repeat, which sounds like a bit of a mouthful, but it's actually referring to a specialized section of DNA. In this section, the bases A, C, G, and T are ordered so that the base sequence is the same if read backwards. Take for example the base sequence G, G, A, T, C, C. Its complementary strand is the same when read backwards, which makes it palindromic. The sequence itself acts as a marker of recognition to identify where the DNA should be cut. CRISPR is part of something called the CRISPR-Cas9 system. Cas9 is an enzyme which cuts the DNA, Cas meaning CRISPR associated. The Cas protein has a guide RNA molecule called sgRNA, or single guide RNA, which unwinds the double helix. CRISPR was discovered in the bacterial immune system as a defense against invading viruses. The bacteria would chop up the DNA of the viruses with the Cas9 enzyme and insert it into its own genome. Then, an RNA molecule would be transcribed from the viral DNA and attached to a Cas9 protein. This forms a sort of immunological memory, so if the virus attacks again, the Cas9 protein with the transcribed RNA can immediately recognize and cut up the virus to prevent infection. In the lab, scientists will design a custom guide RNA that matches the target gene, and they attach it to a Cas9 protein. The RNA then locates the target gene and unwinds the double helix by attaching to the complementary base pairs on the DNA. Once the DNA is unwound, the Cas9 protein cleaves the DNA with its two nuclease domains, one of each strand. It does this by breaking the phosphodiester bonds between nucleotides. Once the DNA is cut, it activates the DNA repair mechanism, which fixes double-stranded breaks. One type of DNA repair is non-homologous end joining. In this repair mechanism, enzymes called nucleases trim the broken or damaged ends of the DNA. Then ligase, another enzyme, rejoins the broken strands. With this process, the trimmed DNA is not recovered, and these missing bases can cause the gene to no longer be viable, and it is turned off. The other DNA repair mechanism is called homology-directed repair. This repair uses template DNA from the sister chromatid to recover any missing DNA from the break. An mRNA complex, which is a complex made of 3 proteins, trims the 5' strands of the DNA, leaving a 3' overhang. The 3' overhang then unwinds the double helix of the template DNA and uses it to fill in the missing nucleotides. Now, all of the missing nucleotides can be filled in, leaving the double-stranded break fully repaired. Homology-directed repair is good because scientists can use a custom template DNA which can be used to edit a gene by changing one or several bases. There are also several ways that scientists can change the Cas9 protein to have different outcomes. One or both of the cutting domains can be turned off so the Cas9 no longer cleaves the DNA. Instead, molecules such as transcriptional activators are attached to the protein. These promote the transcription of the target gene, so more of it is made. On the flip side, factors such as Crab domains can be fused to the Cas9 protein to stop transcription of a target gene. CRISPR can be used to edit the genome of all organisms as the genetic code is universal, so not just humans can benefit from it. In real-world applications, CRISPR has a vast array of potential uses. From treating genetic illnesses like cystic fibrosis, communicable diseases like HIV and malaria, even genetically modifying crops to improve yield and resistance to disease and drought. CRISPR is also being used to prevent rejection from organ transplants and even edit the genome of babies for favourable characteristics. However, CRISPR can be taken too far sometimes, and there are risks that come with the ability to edit genes so simply. It can cause off-target effects in which the wrong section of DNA is cut, leading to unintentional mutations. Also, there is not enough evidence to show that editing the genes of one person won't have adverse effects on the future generations. Nevertheless, CRISPR is undoubtedly a massive jump forward in gene editing and has many potential uses as a promising treatment for a large array of diseases and as a much faster and effective way of gene editing.